Hello everyone, welcome back to this video. Here I am going to talk about how RNA is processed to become a messenger messenger RNA and are to be exported out of the nucleus and turned into a protein. In the previous video, we have discussed about how RNA is transcribed from DNA double helical structure, and those transcribed RNA are called immature pre-mRNA, which cannot directly go for protein synthesis. So for this, pre-mRNA must undergo some processing to form a messenger RNA so that it can help in translation process. So what is RNA processing actually? You may see different definitions about it. In this process, the newly transcribed RNA undergo various alterations to form a messenger RNA. In this process, it requires some modifications like cleavage, splicing of the primary transcript or pre-mRNA. So it's carried out by some nuclease enzymes like RNAs, which cleave the RNA during this process. So if we compare the prokaryotic and eukaryotic gene expression, then we can see a vast difference in between them. In prokaryotes like bacteria, transcription and translation, both of them takes place at the same time, and it occurs in the cytosol. Here you can see in prokaryotes, transcription and translation occurs in the cytosol. This is the prokaryotic cell, the double helical DNA structure which undergo transcription process and it forms the mRNA strand. After that, at the same time, when ribosome get attached to the mRNA, it translates it into protein. This is the polypeptide cell. Okay, but in case of eukaryotic cells, transcription takes place in the nucleus, while translation is in the cytosol. In eukaryotes, you can see here, this is the nucleus, and here are the double helical DNA structure undergo transcription, and this is the RNA processing. Okay, so this is the pre-mRNA. After transcription, it is immature one, and it must undergo some modifications like RNA processing, which involves cleavage, like splicing, etc. Then it becomes mature mRNA. Okay, so that means for translation and for protein synthesis, the mRNA, this mRNA strand, it requires to export it out of the nucleus to the cytoplasm. Okay, so we can say that the protein synthesis takes place outside the nucleus. That means it takes place inside the cytoplasm. This is a eukaryotic cell. After transcription, it undergoes some RNA processing. Then the pre-mRNA undergo modifications to form mature mRNA, and then it becomes exported out of the nucleus through the nuclear pores, and here translation begins. Okay. So for pre-mRNA processing, it includes some events like processing, that is addition of 5' cap and addition of polyadenylation or poly-A tail. Okay. Here we can see a fully mature mRNA, this one, and here is the 5' cap, and here is the 3' tail. Okay. So these are the modifications happens after transcription. So let's see each one individually. So on the 5' end, this is on the 5' end of the mRNA, the pre-mRNA, Once transcription occurs, the 5' end get added with 7-methyloguanosine, this is triphosphate molecule. So once the transcription takes place on the pre-mRNA, the 5' end it get attached with 7-methyloguanosine triphosphate molecule. So it is a modified nucleotide, a kind of upside down in reverse configuration is added to the 5' carbon of the very first nucleotide of the mRNA. The 5' cap actually get added almost immediately once the mRNA is made, after 25 nucleotides. After 25 nucleotides have been transcribed, then immediately those 5' cap get attached with methyloguanosine triphosphate. Okay. So again, in the case of poly-A tail, the 3' poly-A tail, once the mRNA is finally made, there are a series of enzymes which bind to the 3' end of the mRNA, and they will catalyze an addition of a long chain of adenine nucleotide at the end of the mRNA. Okay. So poly-A tail means a long adenine nucleotide chain. Okay. So once the mRNA is transcribed, this 3' end of the mRNA, it binds with a long chain of adenine nucleotide, okay, at the end of the mRNA, and this is known as poly-A tail, addition of poly-A tail, and the phenomenon is known as polyadenylation. Poly-A tail is actually varies in length. It can be 10 nucleotides long or 100 nucleotides. So each mRNA have a different length of poly-A tail. So what is the purpose of this modification? This 5' cap and poly-A tail. So why this is necessary? So there are lots of purposes, but the mainly they are involved in the stability either of the mRNA transcript or translation. For example, the 5' cap is added immediately once the mRNA strand being made. This protects the end of the mRNA from being degraded. So there are lots of enzymes in the cells that can break down any unprotected nucleotide. So this 5' cap protects the transcribed mRNA from being degraded. Similarly, as the mRNA is made and finished, in the addition of poly-A tail also protect their ends of the mRNA from degradation. So both at the end of mRNA get protected by this modification, so it can easily translate it into a protein. The main issue of 5' cap and poly-A tail is recognition and stability of mRNA strand. So this is the post-transcriptional RNA processing. In this case, the bacterial do add 5' cap and poly-A tail, and anything about bacterial mRNA is typically their genes are continuous. Okay, in case of bacteria, the genes their genes are continuous. That means they have uninterrupted coding region. See, in bacteria, you can see the first point. In bacteria, most mRNA contains the uninterrupted coding region. So this is the prokaryotic gene, like bacterial gene. Here, this is a continuous coding region without any interruption. So for example, the DNA has a code for protein. They has the entire sequence of the protein in one continuous stretch of DNA, and therefore When the prokaryotes making its mRNA, they synthesize the mRNA. So we have some ribosomes there and immediately turn into a protein because this coding region is sufficient to turn all this into protein. But in case of eukaryotes, the DNA is different. DNA in eukaryotes is composed of some coding region and some non-coding region. The coding regions are also known as exon and non-coding region is known as intron. So in eukaryotic gene, they have exon and intron by alternatively. So most eukaryotic gene is a combination of exon and intron. Okay? This is the exon, intron, exon and again intron. So this gene is combination of exon and intron, that is coding and non-coding region. So you cannot process non-coding region into translation. So this is a stretch of DNA, suppose, for example, and we want to synthesize protein from this strand. For this, you want to remove those introns. Otherwise, it will stop the translation process because those introns, they are coded for nothing. Okay? They are functionless. Okay? So it may stop the translation process. So the removal of those introns and put together all the exons which actually turn into protein, and this process, the process of doing this is called splicing. This is splicing. Splicing, that means splicing is the removal of non-coding introns of DNA and the splicing together of all the coding enzymes to create an intact mRNA that has a complete coding region that is needed to be made into a protein. So splicing is the third event that happens in pre-mRNA to mRNA, and it takes place in the nucleus. So there are another thing, that is alternative splicing, where all non-coding introns are spliced out by pre-mRNA spliceosome. Okay, so it is a very large complex of protein spliceosome. It recombines the introns and remove them from the pre-mRNA and put together the exons to make a fully final mRNA. So splicing is responsible for making mRNA. This is a massive mRNA structure. This is ready to be turned into protein. So you have your 5' cap and you have your 3' poly A tail, and also you have your fully coding sequence having all the introns removed and only exons. So this is 5' UTR. 5' UTR, that means untranslated region, and this region is 3' UTR, the 3' untranslated region. So these are the spatial non-coding regions. Both these two regions are spatial non-coding regions actually kept at the mature mRNA that help to regulate and how much the RNA has expressed. Okay, so it is clear. This is the mature mRNA strand where you have 5' cap, 3' polyethylene, and a fully mature mRNA strand along with 5' UTR untranslated region and 3' UTR. Okay, so when the fully mature mRNA undergo protein synthesis, it forms a protein structure. Okay, so here is the review. The RNA capping happens at the 5' end of the RNA. So usually it is a 7-methyladenosine. Shortly after RNA polymerase makes the 5' end of the primary transcript, then again splicing takes place, which removes all the introns and join all the exons. Then again polyadenylation, which modifies the 2' end of the primary transcript by addition of a long chain of adenine nucleotide. Okay, so thank you all. I hope you enjoyed the video and hope to see you in the next video.